Hello, and thank you for joining us today as we discuss the development of nanosuspension formulations for poorly soluble drugs. So some of the objectives that we have for today is to discuss the drug bioavailability challenges, potential solutions, wet nanomilling, why, the equipment that we use, and the process that we use for it. So Altasciences, who we are your single source, one-stop solution for early phase drug development outsourcing. We positioned ourselves as a different kind of CRO, CDMO. We like to partner with our clients and focus on creating personal and tailored experiences for our clients and create those successful partnerships that accelerate drug development without increasing risk. So at a glance, we're a mid-sized full-service CRO, CDMO with over 25 years of experience in the industry. We have a strong leadership team. We have sites across US and Canada that we'll see here in a few slides. And these sites have state-of-the-art facilities. We take a fast and easy drug development process approach for a single study or complete program for our clients. We were founded in 1992. We're about 1,200 employees currently. So some of the core values that we have here at Altasciences are integrity, honest and moral principles, quality and excellence, employee development. We provide leadership and really encourage professional development for our employees. Respect, this obviously goes to our employees, but to our clients, no matter what size the client is. And then customer focus. We've built our business listening to our clients and becoming their partners in their development of their API or their dosage solution. So some of the solutions that we do provide here, you can see across the full spectrum from discovery to preclinical into all phases of clinical, and then finally post-clinical market studies and commercial manufacturing. So this CDMO Philadelphia site is represented at the bottom there in purple, the manufacturing and analytical services. We help our clients from preclinical, again, all phases of clinical, and then we grow with our clients into commercial production and manufacturing for them. Here's a list of our facilities. You can start on the left there with Los Angeles is the clinical facility. Up top, Seattle is preclinical and bioanalytical, leading into bioanalytical in Laval. The clinical facilities in Montreal and Kansas City, another preclinical facility just up the road from us in Scranton, and then we're in the lower right-hand corner there for manufacturing and analytical services just north of Philadelphia.
Hi everyone. So thank you for joining us for this webinar today. We want to focus on here is how to improve bioavailability. Low bioavailability has always been a major problem and a key factor in development of any drug products. It has become an ever-increasing issue for modern drug development projects due to the instability of these drugs. It has been estimated that 40% of marketed drugs and 90% of drugs in the drug development pipeline are poorly water-soluble and fall within the biopharmaceutical classification system, Class 2 and Class 4 drugs. As you can see on the chart on the right, these classes of APIs have poor solubility and potentially poor permeability. BCS Class 2 drugs, which is where we're going to focus on today, have poor bioavailability, which is primarily due to poor solubility and dissolution rates. Using these drugs effectively in the clinic requires some enhancement of the drug product through formulation development. And as more and more of these drugs have come into the pipeline, we've seen an increase in NDA approvals for formulations with these enhanced formulation strategies. So new soluble drug delivery technologies have also been employed for lifecycle management of existing drugs and drug products to try to extend patents, improve activity, and create value for companies who take that approach. So there's a number of common strategies used to improve drug distribution and bioavailability of poorly soluble drugs. The first technique I will go over is the use of co-solvents. Using co-solvents is a simple technique that is fairly low cost and is applicable to a wide range of drugs. However, there are several disadvantages that often steer people away from this approach. One is the toxicity of the solvents that are often required. There's also the risk of drug precipitation once the drug is in vivo and in an aqueous environment. There's a tendency for the solvent to diffuse away from the drug, causing it to precipitate, thus causing you to lose the advantage that you were achieved by the use of the solvent in the first place. And finally, it's limited to liquid formulations only. It's not a strategy that can be used for oral pills tablets, capsules, as well as freeze-dried products. Another approach is compensation using cyclodextrin, as cyclodextrin is used to improve chemical stability of the drug via inclusion complex with the API, and thus may potentially enhance the drug absorption. However, success depends largely on being able to match both chemical and geometrical structures or properties of the drug molecule with the available cyclodextrin. Occasionally, large amounts of cyclodextrin may be required if the inclusion complex and the complication efficiencies are poor, thus leading to higher costs for each excipient for solving these bioavailability problems. Another approach is the creation of solid dispersions. This can be done by spray drying the API, either solubilized or suspended with another agent that will help encapsulate and stabilize. This creates fine drug particles without excessive application of energy that are readily available with minimal risk for agglomeration or aggregation. Another approach to these solid dispersions is that there are a wide range of hydrophilic polymers available as drug carriers, so this is a highly used approach for increasing bioavailability. But again, there are some disadvantages. For example, the preparation method is often difficult to scale up, and there are not many sites that have that availability. For example, commercial spray drying equipment is another disadvantage that creates amorphous drug forms which are physically unstable and can convert to the crystalline form during storage. This can change the stability, change the bioavailability, and change the shelf life available to these formulations. Another approach is that it's often in the news today with the COVID vaccines is the lipid formulations. These exploit the innate lipid digestion mechanisms of the body which enhance the drug's bioavailability. These emulsify lipid formulations further enhance lipid digestion and therefore drug availability. There is a diversity of lipid excipients that allow the formulators to have a lot of flexibility during developing or in developing these formulations. And there's a lower risk of drug precipitation in vivo because these particles tend to stay together in an aqueous environment. This is all due to the stability applied by the lipids. Again, disadvantages in this case can be the cost. Cost can become a factor as there's often 2, 3, and sometimes as many as 4 different lipids involved in the formation in these formulations. High drug load can also be difficult to achieve, thus increasing the cost and sometimes eliminating the usefulness of these formulations. And then finally, quality control of the lipid-based formulations is complex, having to characterize not only the API and its stability, but the lipids and their chemical and physical stability as well. Where do we start? As we discussed on the previous slide, for BCS Class 2 drugs, the dissolution step is the rate determining factor in drug absorption. Current strategies employed to improve the apparent solubility of a drug include the use of co-solvents or for example, low molecular weight polyethylene glycols and propylene glycols in combination with water to dissolve the drug, complexing agents such as cyclodextrins and its derivatives to form water-soluble inclusion complexes for the drug, hydrophilic excipients. This is your PVAs and your other high molecular weight polyethylene glycols. Hydrophilic excipients act as drug carriers for the preparation of solid dispersions in which the drug is dispersed molecularly or as ultrafine crystals. And finally, lipid formulations such as solid lipid particles or liposomes. We take a different approach. We try to increase the rate of dissolution step by decreasing the particle size through nanomilling. Nanomilled particle size reduction, aka milling, is a more general method where mechanical energy is applied to physically break down coarse particles into finer ones. So I want to go through some of the benefits of nanomilling and why we do it, how it's done, and how it's been used by others. So I promise this is going to be the only math you're going to see today, and that is the Noyes-Whitney equation. Essentially, what this says is that the rate of dissolution is dependent on a number of factors we have no control over, such as diffusivity, the thickness of the aqueous boundary layer, the concentration of the solute that's dissolved, and the equilibrium solubility of that API. The one thing we do have control over is that we can control the surface area for dissolution. To break it down and eliminate the math, the smaller the particle size, the greater the surface area. The greater the surface area, the faster the rate of dissolution. The faster the rate of dissolution, the faster the drug absorption. As you can see on the right, as the particle size decreases from 10 microns to 1 micron to 0.1 microns, there is a noticeable and exponential increase in the surface area, also which leads to a dramatic increase in solubility and bioavailability. So I have an example of what I'm saying on the left-hand side of this slide. It is an example of phenylfibrate using as received drug components and versus nanomilled suspension. The as received drug substance, which was in the tens of microns range, only has a solubility of about 10% after 2 hours in standard dissolution. However, the nanomilled drug, which is now down to hundreds of nanometers in size, is 70% dissolved in only about 30 minutes. So that is an increase in the dissolution rate, which translates directly to bioavailability for that compound. And in fact, if you look on the right here at a BCS squares, I've circled a number of compounds that have benefited from nanomilling. By decreasing their particle size, you increase their surface area, thus their bioavailability. Nanomilling is defined as a unit operation where mechanical energy is applied to physically break down coarse particles into finer ones. Why do we like nanomilling so much? We like it because virtually every drug can be ground into finer particles by adding energy, and that goes for the aqueous and non-aqueous soluble compounds. There have been a number of technological advancements that have helped increase the scale of the production and stabilization of ultrafine particles, thereby increasing the surface area, increasing dissolution rates, and making possible the delivery of compounds that previously were considered brick dust and not good as development candidates. There's a number of FDA-approved animal drug products on the market. I've listed 12 of them here. And what I would like to point out is that they come in a variety of dosage forms. For example, some come in tablets, capsules for oral delivery, there's oral suspensions, but also injectables for intramuscular and even IV. So nanomilling brings into the realm of possibility a number of delivery formats for your drug, depending on what your needs are. So as for the other delivery methods that we talked about earlier, there are issues for decreasing particle size. One is that when you're milling, you're taking larger particles and increasing the surface area of those particles by making them smaller. You are therefore increasing the free energy and decreasing the thermodynamic stability of those particles. There is promotion of particle agglomeration. So with the increase in surface area, there is an increase in the possibility of van der Waals and electrostatic interactions that increase agglomeration. To solve for this, the drugs are co-milled with additives. These additives are inert, non-toxic pharmaceutical excipients accepted by the FDA that serve as a carrier or a stabilizer of the drug. These excipients need to be carefully selected for their delivery method that also allow the formulation to come to a final stable form. These excipients are hydrophilic and confer hydrophobicity to hydrophobic drug particles, therefore enhancing their wetting ability, their solubility, and their bioavailability. There are a number of examples of these stabilizers that are used for some very common drugs, and rather than go through all of them, I just want to point out the variety of the excipients that have been used, that have been investigated to stabilize these drug formulations. So where do we start at Altasciences? We like to be focused and fast. We want to find out quickly if a drug is a good candidate for nanomilling. So our approach is to screen very quickly with inexpensive equipment, excipients, and small amounts of drug. The number of conditions that would be necessary to come to a stable formulation. The process that would be used for screening is easily transferable to scale up using excipients that are GRAS or generally regarded as safe and acceptable for further development. Characterization utilizes easily employed methods, and I would like to stress again, we like to take this approach as fast and focused. So, for the equipment, It's very simple where we start. It's simply a roller mill, often referred to as a jar mill. This is sufficient to screen multiple stabilizers, concentrations of drug and media, the type of milling media, and the amount of time it takes to mill the material. Volumes can be as low as 50 milliliters, and drug amounts can be as low as 1 gram and perhaps up to 10 grams for multiple conditions. So how does this work to reduce the particle size? Well, the figure on the bottom right shows a cutaway of the inside of a jar. It's filled with milling media, your drug, a steric stabilizer, and sometimes an electronic stabilizer or electrostatic stabilizer. And the jar is rolled on the rollers. Picture of this is on the top left of the screen. The media inside the jar, while it's rolling, falls onto the API after rolling up the walls, and crushes the API over and over and over again. So you go from a nice micron-sized API particle to viewed on the left to a nano-sized drug on the right. So the milling media. There are two choices here. One is the ceramic grinding media. This is the most commonly used form is the high-density stabilized zirconia, also known as YTZ media. This provides a greater impact force because it's very heavy and dense material, giving it excellent grinding efficiency. They're available in very high purity with smooth surface and almost perfect roundness. These are important for reproducibility, that we have the same millimeter every time we do a screen or scale up to our production scales. The smooth surface allows for efficient recovery of the nanometer drug, and finally, they have high wear resistance and fracture toughness. So no material from the YTZ media can be found in the final product. The second option is a polymeric milling media known as this is a copolymer of styrofoam styrene that has been cross-linked. It's very tough, not abrasive, and thereby minimizes the wear on your milling chambers. It's sterilized and able to be thermally stable up to 500 degrees in case sterilization is necessary. It's compatible with both oil and water, as the aqueous solution needs to be aqueous for milling. And then also it's environmentally safe and non-porous. Finally, the spherical and narrow particle size distribution that milling gives you is reliable, reproducible for many products. So again, these are the typical stabilizers that are often used. As you can see, there are small molecules like vitamin ETBGS, which is a steric stabilizer and contains long peg chains as part of the molecule. There are polymers such as pluronic F68, methyl cellulose, and then charged molecules like sodium deoxycholate to provide electrostatic stabilization. So why do we need to use the stabilizers? Well, as we said before, aggregation is a problem. As some of the larger drug molecules are milled smaller and smaller, the surface area increases exponentially. Because of this, there is a much greater tendency for aggregation and agglomeration, essentially the sticking together of these particles. It can be so bad that you could potentially lose all the advantages of nanomilling altogether. There's also a second process called Oswald ripening. This is where smaller particles in suspensions dissolve and deposit onto larger particles. Remember that although you have a saturated solution with particles suspended, it's still an active situation where a drug is dissolving and reprecipitating during both milling and during storage. So Oswald ripening is a serious problem that can, in time, even within small increments of time, within hours of milling or long-term storage, affect the stability of these drug particles. What you see after milling is the particle size distribution will start to shift. So the black on the bottom right-hand chart is an example of where you might be after milling. This is a good starting point for your stability. But within 1 hour, 1 day, the particle size distribution has shifted to the right. Particles are getting larger, and that's going to decrease the solubility rate of your formulation. So the next step in developing the screening is choosing the drug concentration. Things to consider are what's the final concentration of the drug that you want in your final formulation. Higher concentrations may require higher stabilizer concentrations. So the goal may not be to get as high as you can, but get to some level where it's efficient. You have enough concentration to further formulate to your final formulation and use a reasonable amount of stabilizer. The amount of stabilizer is important because there will be an increased need to increase the amount of stabilizer during the time, during the milling, and may take you above a safe level for use of these excipients in a formulation. Finally, high concentrations of drug and stabilizer may actually decrease the milling efficiency by increasing the system's viscosity and decreasing the collision energy. The next step is to select the milling media size and concentration. So typically we would like to start with the YTZ media. A number of sizes are available. We normally start with a 0.5 millimeter or perhaps a 1 millimeter with a loading of about 50% for very small target particle sizes, say 200 nanometers or less. For the 200 nanometers or less particle size, we would need to use smaller milling media, which are also available. Then you need to select your stabilizer. So it's important to consider that the expected delivery method when choosing your stabilizers. For example, there are few GRAS stabilizers available for injectables, so really your choices are limited. There are, however, many stabilizers available for orally administered drugs, and choosing the right formulations to the right stabilizers to start with will add to the speed of the quality of the results. Next is the roller speed. Remember, this is a jar that is spinning, so if it spins too fast, the media and the drug will simply stick through centripetal force to the outside of the inside surface of the jar and not reduce the particle size very quickly. You want these particles to interact with the media, having the media fall on them, and so you want to have the speed of the jar appropriately selected so that this happens. Finally, you need to measure the particle size in real time. On the right, typically what you would see on the right is how this would play out. Starting on the right is the large initial distribution, and as time goes on, you would see the particle size distribution shift to the left. It gets pretty ugly there in the middle as the particle size distribution gets very wide and disorganized, but as you get towards the end of the milling process, it should all come together in a nice bell-shaped curve distribution. So the equipment for this, as we showed before, is a simple roller mill, which can handle a few stabilizers and conditions. It can handle roughly 3 or 4 formulations per piece of equipment. We do have a wide choice of volumes and types of containers that you can use on this mill. However, you may have a more aggressive timeline. So instead of a few stabilizers, you might want to look at a slew of stabilizers and conditions. This is another roller mill that can handle maybe 9 to 12 different formulations and conditions all running in parallel. And finally, if you need a larger volume, larger scale, simply buy a larger jar. And these are readily available as well, essentially using the same equipment for the screen. So where do we go from here? We need to choose a characterization method. There are 2 main characterization methods for this. One is particle size distribution by laser diffraction. This is good for anywhere between 10 nanometers and 5 millimeters. This can monitor the wide range that you'll be measuring over the time course of the milling. It's great for suspensions, powders, paste, gels, and creams. But of course, we're looking at suspensions here. It's flexible, fully automated, gives you maximum precision, and it's also compliant for the FDA. This equipment gives excellent reproducibility and agreement from equipment to equipment. As for the measurements, they're fairly quick, in say about 1 to 2 minutes. On the bottom left, you can see a typical before and after. The green is a typical particle size distribution of, let's say, jet milled material that would normally come in. In the red is the nano-sized particle distribution typically get from milling. The second approach is by dynamic light scattering. This is best for smaller particles. Particles, let's say, between 1 nanometer, 1 micron, and perhaps up to 10 microns. They're simple, no sample preparation, as this can handle high concentrations that come directly out of the nanomilling process. It can also handle turbulent samples that need to be measured directly. The mean size only determination requires knowledge of the viscosity of the liquid, and then measurements are done in less than 60 seconds. So what do you want to look for when you're checking the particle size distribution? Is a general characterization that is often used for these types of measurements. That's not only the measure of the median, which is determined or called the D50, but also the D10 and the D90. The D90 is the particle size where 90% of the particles are below this size. This is particularly important for injectables, as anything being injected IV will have a limit on the upper end of the size of the particles that can be injected safely. The other is the 10%, also known as the D10, which is the number of particles that are below 10% of this particle size. And again, these give you a general description of the shape of the bell curve, which can be different for every molecule, different milling times, and also different stabilizers. And finally, as we mentioned before, you want to check for aggregation and Oswald ripening. You may want to take your milled material with the stabilizers and let it sit for a while and measure the particle size over hours and up to over a couple days, just to make sure there's a physical stability there before you move forward with any further formulation development. You also want to look at the dissolution rate. As we saw before, decreasing the particle size exponentially increases the dissolution rate. So you want to check various media. This can be done by comparing it with or checking it with standard buffers that are used for dissolution of oral fed simulated media and other simulated media, depending on where the delivery will occur. For example, saliva, vaginal fluid, and even vitreous humor. So a number of these are available and can be selected specific to your delivery method that you're interested in. Simply take those samples as they dissolve on the time scale, inject them into an HPLC, and get a dissolution rate as you can see on the right. So where do we go next? Well, you're going to want to scale this up once you determine what's a good formulation stabilizing drug combination. Here at Altasciences, we use a delta vita. We have a pilot scale delta vita 300, which is good for early stage clinical manufacturing, as well as any preclinical work. And then we have the delta vita 600, which we use for larger, later stage clinical and commercial scale manufacturing. So just to give you an idea of what these look like, we've done a cutaway of a general diagram of these sorts of machines. On the left, a drug suspension which is stirred and pumped into a chamber with milling media. It also contains obviously the drug and the stabilizer. A blow up of that, as you can see, it's a spinning agitator with pegs that essentially move the milling media and the drug together. That's the energy input. And then this can be either collected or can be recycled, as you can run several passes of the suspension in order to get the particle size down to your desired target. So I guess the message is here that you don't have to imagine necessarily what your drug product is going to look like. Only what properties you want. Leave it to us to outsource the rest. Outsource to us to do early phase drug development in cooperation with our preclinical and clinical sites within Altasciences. Again, there are a number of sites available across North America. Our site here in Charlottesville, Pennsylvania is just north of Philadelphia, and in the pharmaceutical and biopharmaceutical arenas in New Jersey. And now some questions. If there are any questions from the audience, I'll be happy to answer them.
Yes, looks like a few questions have come in. So I'll just read the first couple questions here and give you an opportunity to respond. So the first one is, does scaling from a roller mill to high energy media mill like a Delta Vita require much formulation or process optimization?
It's actually less than you might expect. Typically, the media loading and the stabilizer concentrations would be unchanged. There may be some small tweaks required as you go to a larger scale up with the particular incipient level. The most dramatic changes would be determining the milling time. Since it's a much higher energy method, sometimes those milling times are extremely short, shorter than you might expect, and there's a danger of overmilling, which can actually start to increase particle size and change from the crystalline form of the API. So really, milling time is the only factor that we need to pay attention to when you move to high energy equipment.
Okay, thank you. Next question. Where can we look to confirm that a stabilizer is considered suitable by the FDA?
Well, the FDA on their website, on their public website, has an inactive ingredient database. Essentially, it's a list of all the drugs that have been marketed with all of their excipients, and even in many cases, the levels and amounts of those excipients that are in those products. That is the primary place we look to select among the many excipients and stabilizers that are available. It will also tell you whether those have been used as oral, injectable, topical, or say mucosal stabilizers or excipients. So that's where we would want to look first if you go to an FDA inactive ingredient website. The FDA website, and search for their inactive ingredient database. It'll pop right up to the top.
Wonderful. How can the nanosuspensions be further processed for other dosage forms?
Well, there's a number of different ways. Typically, you're milling at a very high concentration, so they're often diluted for oral suspensions or eye drops. They can be localized. That can add additional stability, physical and chemical stability to those formulations. They can be absorbed onto another particle, like a silica particle, to help with delivery, and then incorporate it, perhaps into an oral solid dosage form. They can be spray dried, so there's a number of different ways that they can be further processed and to develop into a final formulation.
Okay. So we'll just field two more questions here. So can potent compounds be nanomilled at Altasciences?
They can. We have set ourselves up to handle potent compounds in another number of formats and with a number of processes, including nanomilling. So yes, absolutely. There is an availability that's available here at Altasciences.
Okay. And final one we'll take right now is, how small can nanomilling take an API down?
Well, that's a great question. That's one that we're asked very often, and the answer is it depends on the API itself. The friability, fracturability of those API crystals, which can depend on the crystalline form as well as other salt forms of those drugs. It's the most likely determining factor, given that particles can be nanomilled and have been nanomilled to generally easily between 100 and 200 nanometers, and sometimes even lower. Well, I've seen some down to 50 nanometers for certain drugs.
So I think that's all the time that we have right now for questions. And please feel free to reach out to any one of us in this presentation to continue to ask questions about Altasciences or nanomilling as a whole. We'd like to say thank you one more time for joining us today, and wish everybody a good rest of their day.